about linear deconvolution of DNA.
Thank you. I think that's good. Oh, I can. Okay. It's going to go over here, full screen. Okay. Okay. Thank you very much. It's a pleasure for me to be here. Is this okay? With the okay. As you might be able to tell from the catchy title of my talk, I'm going to be assuming some basic familiarity with mass spec proteomics. That said, I think many of us in the room are aware that there's a serious problem with reproducibility of mass spec data in a typical discovery proteomics workflow. And that's because the generally taken approach, which is data-dependent acquisition, or DDA, intrinsically involves a stochastic element of the data gathering process. So as your sample is entering the instrument via liquid chromatography, the mass spectrometer takes a mass spectrum, an MS1 scan, just to get a census of what's present at the moment every couple of milliseconds. And within that MS1 scan, it then proceeds to sequentially isolate individual peptide precursors for fragmentation to obtain MS2 spectra specific to that precursor, which can then be used for sequencing. And it then continues to do this for only a couple of the most abundant precursors that are present in that MS1 spectrum, and then it moves on, takes another MS1, and the process continues. So that means that the less abundant species within your sample, not necessarily particularly low abundance, just anything that's not among the most abundant, will never be seen via this approach unless you have some a priori kind of targeting method, as we saw before. And this leads to serious problems with reproducibility, where even with technical replicates of the same sample, we can have a situation where, due to natural technical variability, a particular precursor is abundant enough to be sequenced in replicate 1, but not in replicate 2. And so you end up with data matrices that have horrific numbers of missing values, as indicated by the black spots in this figure from a recent study. And this is particularly problematic for something like clinical applications where you have dozens or hundreds of samples that you want to interrogate simultaneously, and making some kind of biological inference about the roles of certain proteins in certain diseases becomes very confusing if you have so much missing data. So data-independent acquisition, or DIA, was conceived as an alternative to DDA largely in order to skirt this issue with non-reproducibility. And the way it does it is by eliminating this element of stochastic precursor selection. So rather than taking an MS1 scan and selecting out only the most abundant precursors one at a time, in DIA, we select entire groups of precursors to be fragmented simultaneously. And they're selected based on the criterion that their mass-to-charge ratios should fall into a relatively wide window. Everything in that window is simultaneously fragmented, and that window is then ratcheted along the entire mass-to-charge range so that we obtain, at least theoretically, a more or less complete record of everything that's observable by mass spec within our sample. And as you can imagine, the data matrices we obtain from this approach are significantly more complete than their DDA counterparts. Still not perfect, but the situation is much better. But of course, we've paid a price to do this, which is that the MS2 spectra we're acquiring in DIA are now convoluted spectra. There's no longer a direct relationship between MS2 spectrum and peptide sequence. You have to do something in order to extract the information that you want from these spectra, and applying typical DDA database search type tools will simply just fail with this kind of data, so you need new tools. And the most common approach, probably the main approach, really the only approach that's been taken up to this point in analyzing these kinds of data, is to take advantage of the retention time axis of information. So in DIA, because there's none of this individual selection of precursors, there's no kind of dynamic exclusion. So as a precursor is eluting off the liquid chromatography, you get multiple points along that elution profile where it's being sampled. So you can try to look for a particular precursor by taking some assay of diagnostic fragment ions associated with that precursor, and looking for correlations between the chromatographic elution between those fragment ions at the MS2 level. And theoretically that works well, and sometimes it does work quite well in practice, but often in practice, because we have all of this co-isolation of precursors in these wide DIA windows, we have these interferences. So rather than getting nice, clean correlation profiles like this, you end up with something more like this. So you have to start introducing sort of heuristic rules in order to deal with such situations. And at the end of the day, you start including certain things, throwing other things out, and it's not clear how much of what you're identifying and quantifying is really reflecting what's there versus being more technical artifact. So we wanted to develop an algorithm that would allow us to analyze these data directly without making use of the retention time elements, not looking for correlations between fragment ion elution profiles. And we call this algorithm Spectre. It appeared in Nature Methods earlier this year. And it's based on a very simple hypothesis, which is that any of these convoluted DIA MS2 spectra are no more, no less than just a linear combination of the DDA type sort of pure spectra associated to each of the precursors that were co-fragmented to acquire this DIA spectrum. And what this is saying in simple English is that the number of fragment ions with a particular mass to charge ratio in one of these convoluted spectra is simply the sum of the number of ions contributed by each of the precursors that were co-fragmented when you made that spectrum, along with some noise and some error. So this takes the form of just a familiar matrix equation, the most basic equation from linear algebra, plus an error term. This is a targeted approach, meaning we need to begin with a spectral library. It can be quite large. So we begin with a spectral library that defines the space of peptides or precursors that we'll be able to actually identify in this approach, because we need something to deconvolute onto. And this can be efficiently solved with non-negative least squares. The non-negativity is because the abundance of any particular precursor can't be a negative number. And by solving the system, we end up with this vector of coefficients, one coefficient for each precursor in the library, that tells us what's the total ion intensity of that particular precursor within this particular acquired MS2 spectrum. So there's no retention time element. This is at a spectrum by spectrum level. But you can imagine for any particular precursor, you get a sequence of coefficients, one for each of these MS2 spectra, and that gives you a kind of calculated total ion chromatogram. So we wanted to test what are sort of the new features of this method. What advantages does this give us that pre-existing tools didn't have? And first of all, there's the notion that DIA should give us very sensitive quantification at the MS2 level, which we assess by spiking a panel of 96 synthetic phosphopeptides into a non-phospho-enriched background, so endogenously you wouldn't see them without the phospho-enrichment, spiking them in along a linear 5-point gradient, analyzing that mixture, that spike in mixture via DIA, and then analyzing the data either manually in Skyline or automatically using this algorithm. And what we found was that both qualitatively and quantitatively, in terms of what Spectre was identifying and what it was quantifying, it agreed with manual annotation extremely well. And it gives you this point-by-point calculated chromatogram that agrees very well with what you would find manually. That was just sort of a basic sanity check. I wouldn't call that something novel. The real novelty lies, I would say, in two sort of features that we highlight in the paper. So the first is that this approach, this linear deconvolution approach, comes with an intrinsically low false discovery rate. You're very unlikely to say that something is in the sample if it is not actually there. And that's because the linear algebra framework is very unlikely to assign positive coefficients to precursors that are not well represented in the data. That tends to increase the error rather than decrease it. And we assess this simply by taking an on-target library, which we'd used to analyze the previous data set, the spiked-in data set, taking that library and augmenting it with a whole bunch of false positives, a whole bunch of precursors which we knew were not there because they came from E. coli. And we simply reanalyzed the data using this much larger sort of hybrid library and found that Spectre essentially identified none of the E. coli precursors while identifying a substantial proportion of the significantly smaller on-target library. So you can still do some a posteriori kind of false discovery rate control using a typical target decoy kind of approach, which we do eventually do to reduce the false discovery rate further. But false identifications with this method are really much less of a concern than has been the case in DIA so far. Now the next novel aspect of this approach is that it's extremely sensitive. So it's able to distinguish between peptides with highly similar sequences, things like peptides that differ only by substitution of a single amino acid or maybe transposition of adjacent pair of amino acids. And this is a scenario that's been very difficult to deal with in DIA so far because the data is already so complex due to this co-isolation of multiple precursors. Looking for that level of specificity is just been too refined a problem to examine in DIA so far. So to test Spectre's ability to do this, we actually synthesized families of peptides with highly similar sequences. As you can see here, here's a family of 6 where they only differ from each other either by swapping adjacent amino acids or, in the first case, substituting a leucine with a valine. And we then mixed together random subsets of these family members and spiked them into an E. coli lysate and ran those whole mixtures in DIA and used Spectre to analyze them. And what we found was that in each case, with each of these mixtures, here's a mixture of 2 of the members of the family, here's a mixture of 4, and here's a mixture of all 6 together, Spectre correctly identified the precursors that had in fact been mixed together, and it did not identify any of the other members of the family. So both the specificity and the sensitivity were pretty perfect, in at least for this particular case. And to compare this to what we would have gotten more likely using pre-existing approaches, we calculated a more typical kind of measure of spectral similarity than this kind of linear algebra deconvolution coefficient thing. A more typical measure of spectral similarity would be something like the normalized dot product. So we calculated also the normalized dot products between the spectra of these synthetic precursors and the acquired DIA MS2 spectra. And what we see is that the normalized dot product is just completely unable to distinguish between these, which is more or less what we would expect. So this level of sensitivity is pretty unprecedented. So circling back to my initial point, I want to say, what is it that we actually get by having this improved sensitivity coming from the linear deconvolution together with the reproducibility that's sort of being afforded by the DIA approach? So we have we gather a lot of phosphoproteomics data sets in our lab, and among them we have subjected a standard cancer cell line, PC3 cells, to a wide panel of kinase inhibitors, so 32 different kinase inhibitors, and ran each of those samples with biological replicates in DIA. And among that DIA data, so we end up with about 96 samples in total, including replicates. And within those 96 samples, we found this particular pair of positional isomers, meaning the same peptide sequence, but just phosphorylated at different serine residues within the sequence. This particular pair of positional isomers, both of them were identifiable in the vast majority, something like 88 out of those 96 samples. So that's kind of the reproducibility. But it's also exploiting the sensitivity because these positional isomers are really hard to distinguish in DIA. Their spectra are very similar. Their sequences are similar. And as you can see here, they nearly coelute in most of these conditions. And by this level of sensitivity allowed us to formulate a new biological hypothesis. This peptide is a constituent of the C-terminal domain of the membrane cross-linking protein plectin. And it's been known that plectin is phosphorylated somewhere in the C-terminal domain by these 2 kinases, CDK1 and MNK2, but it hadn't been clear precisely where each of those kinases acts. And using this level of sensitivity together with being able to observe the patterns of relative phosphorylation of the 2 positional isomers across this panel of kinase inhibitors, we could infer where precisely, at exactly which serine residue each of these kinases phosphorylates plectin. So with that, I will simply say thank you to everyone here, thank you to my redoubtable colleagues and collaborators, and I'll take any questions you might have.
So this is great. I think it's really interesting. One question is, in any kind of regression model like this, all the stuff that's not in your library, which corresponds both to noise but also to peptides that you didn't happen to have a library for, or all the unknown peptidoforms, gets lumped into that E term.
Yes.
Which can be problematic, especially if you don't have very good coverage in your library.
Certainly.
I wonder whether you've done any kind of downsampling of the libraries to investigate this. So if you toss out some of the stuff from the library, do the detections of the things left in there suffer because the E term has to account for all those unmatched signals?
So we did, in fact, do that downsampling. It's in a supplemental figure in the paper. I don't have it here. We did it in the context of this LFQ bench data set, which involved mixing together the proteomes of 3 different species, E. coli, yeast, and human, and then running those mixtures in DIA. So we downsampled. We took a library that started with all 3 species and then downsampled only the E. coli precursors. And we found that, you're still accurately quantifying those E. coli precursors as you begin with, start adding smaller or larger and larger proportions of the other 2 species as well. So it was something we were concerned about. And theoretically, I felt that there's a lot of regularity in this regression because of both the non-negativity and the relative sparsity. You expect there to be very few members of the spectral library that get a non-negative coefficient in any particular one of these systems of equations. And so the regularity was sort of the theoretical hope, give us the hope that error term would not be too egregious when things are missing from the library. And then we do have the supplemental analysis. Yeah.
Do you know if the assumption of linearity covers or is true across the entirety of the dynamic range of the vector? Because my understanding is there's potential space charge effects for Burby trap if you're filling it pretty full. I was curious if that's something you've looked at.
Not explicitly. I mean, I've considered sort of, because when you have the error term in the linear regression model, you're making a certain assumption about the Gaussian nature of the biochemical noise. So I think what you're saying could be incorporated by, making certain assumptions about maybe the heterogeneous nature of the noise and might not uniformly affect each of the peaks within the spectrum. Those sorts of mass spectrometric effects. We didn't incorporate that at all, but it could hypothetically be brought into the error term. I actually did a little bit with that, but it just gets so complicated, and the advantage is relatively slim. So.
Very convincing in terms of the identification you're getting. I'm just wondering if you also looked at the accuracy of quantification by using the whole spectrum rather than just single transitions or multiple transitions.
We have assessed that. I don't have an example on hand, but we did consider, again, in the context of that LFQ bench data set, there were 2 spectral libraries that came with the paper, one of just pre-selected assays of about 6 fragment ions per precursor versus the whole shebang. And we found that we actually get substantially better results using the entire spectrum, at least on a Gestalt level of quantifying every peptide within the species and looking at the ratios between different species. I'm not sure on a precursor by precursor basis, I couldn't say, but it's my feeling that using more information here is better. More is better.
I get the question. So how many spectra do we actually need to put in? So how many potential coefficients do we have in
Like how large does the library need to be?
always against the full library, or so how do you identify that? How large are your windows? It's probably based on window.
Oh, yes. So for each window, for each MS2 spectrum, we subset the library to only consider precursors whose mass to charge actually fall into the window. But within that, we've used libraries ranging from tens of precursors to tens of thousands, hundreds of thousands. It doesn't seem to, connecting to the previous question, the size of the library is surprisingly less of an issue than you would think. Kind of as soon as something's there, it tends to be well identified and well quantified with this method, unless there are a lot of other members of the library that look very similar to it, and then you take those out. That can conflate the analysis.
Further questions?
So your model is very simple, I think. If you consider not a peptide but a protein, the peptide from different proteins should be the same level if you include this factor into your model. Do you think it will improve the quantification or just add more noise?
The same. Well, I mean, if they would have correlated quantifications across samples, presumably, but they wouldn't be exactly the same because of the differential ionization efficiencies. I mean, I think there are kind of different layers of the analysis. There's you could try to deconvolute different proteoforms, for example, using a linear model. I don't think that's what you're asking. No, I haven't looking for. Connecting different peptides from the same protein, there's no explicit logic of that in this model. I'm not sure how it would be incorporated exactly.
All right. Thank you very much.
Thanks.